This is podcast number 23. So you know from previous classes that DNA replication is semi-conservative. From two original strands, each strand in dark blue here acts as a template to make a new strand, so that every new strand is half old and half new. Okay? So it's not that two strands make two brand new strands. The two strands, original strands, unwind, and then each strand acts like a template to make a new strand. That's called semi-conservative. And we can see in replicating DNA under a micrograph a fork forming where you have the two strands separating to act as template to form new strands. And in the fork, you're going to see that each new DNA strand has both what's called a leading strand and a lagging strand. And both are made at the same time, but the leading strand doesn't need to be made piecemeal in small pieces called Okazaki fragments. It can be made all at once. So if you have your original strand here in dark blue, 5' on your left, 3' on your right, and then on the bottom strand here, 5' on the right, 3' on the left. When this strand opens up to start being replicated, it's going to be the new strand will be complementary to the existing dark blue strand. So for the bottom, this means that the 5' end will be complementary to the 3' of the original strand, and that new nucleotides can only be added onto the 3' end. So this is the end that is growing, the one with the arrow there. And as this fork opens up in this direction, okay, this you'll just keep on adding to the 3' end. And since you don't have to start over again, this is called the leading strand. On the other end, you have the opposite orientation. So this right here is the 3' end of this top blue strand. And so the replicated strand has to be anti-parallel to that. So that means this end will be 3' and it will, this end right here will be 5' of the replicated strand, and it'll add on nucleotides in this direction. Well, as it first started opening up, you laid down a 5' end and you added on to the 3' end until you ran out of DNA, and then you have to back up and do it again. You lay down a 5' end and replicate DNA until you run into that first strand, and then you do it again. You lay down a 5' end and you make DNA until you run into that second strand. The fragment rather. So these little bits are called Okazaki fragments, and it's a function of the direction that the DNA is opening and what end is complementary to the other end. So in previous classes, at least when I teach this, I say only draw forks because bubbles are more complicated, and in this class we're going to do forks and bubbles. So I'm going to explain this in the slide, and then I will show it to you drawing it out, and then everybody will get it. And it's safe to say that this is something that I'm going to ask you to do on an extended response question. So bubbles are more reality in terms of how things happen in real DNA, and a fork is just half a bubble. So if I extend this to the other side, you can see that I would get a bubble. Okay, if I just add on to the other side. Or if you put your hand over half of this slide, this half here would be a fork going in this direction, this half here would be a fork going in that direction. And that's what makes bubbles more complicated is that the direction that the original template strand is opening is in two directions instead of only one direction. So in a fork, it's only opening in this direction, off to your right. But in a bubble, this side is going off to your right and opening in off to your right, and this side is opening going off to your left. And the direction in which the original double-stranded DNA is opening is key to figuring out what is the leading strand and what is the lagging strand. Okay, I'll say that again. The direction that the original DNA strand is opening, if it's opening to the right or opening to the left, makes a difference in terms of what will be the leading strand and what will be the lagging strand. Okay, why? Let's take just this half of the bubble. It is opening up in this direction, off to the left. So the original strand is 5' on this end, 3' on this end, so that means right down here in the middle of this bubble, that's the 3' end of this original strand. And I'm going to make DNA that's complementary to this original dark blue strand. And so I need to make DNA that is being added on to the 3' end in this direction. Okay, so the original strand is 5' on the left, 3' on the right. The replicated strand will be 5' on the right, 3' on the left, because it's anti-parallel. The red means that you're gonna start off with an RNA primer. We'll talk about that soon. And then that RNA primer provides a place for DNA polymerase to latch onto, and DNA polymerase starts adding on to the 3' end, which is here. And then as this bubble opens up in this direction, it just keeps on adding to the 3' end. And since I don't have to start over and over and over again, this is considered the leading strand. If we look at the other part of this half of the bubble, the one on the left-hand side of the screen, you have anti-parallel original strand, 3' on the left, 5' on the right. That means that the DNA that I'm going to synthesize is going to be anti-parallel to that, 5' on the left, 3' on the right. It is opening up in this direction, okay? And if this is the 5' end, I'm going to add nucleotides to the 3' end until I bump into another fragment and another fragment. So this was the original fragment as the bubble first opened, and I added until I ran out of room, bumped into another fragment up here, and then I had to back up as the bubble opened more, in this direction to the left, and I made another fragment that connects with this fragment. And then as the bubble opens up more, I made another fragment opening up to this fragment. These fragments are called Okazaki fragments, and because it involves more steps in that you're making DNA in small little bits, and then later on you are going to ligate all these bits together. You're going to replace the RNA primers with DNA, and then you're going to ligate all these bits together so you get one strand of DNA. That takes more steps, and so that's called the lagging strand. Okay, so that's on this half of the bubble. On the other half of the bubble, it's opening up in the opposite direction, and because it's opening up in the opposite direction, that means what is the leading strand and what is the lagging strand will be different than it is when it's opening up to the left. Now it's opening up to the right. Because it's opening up to the right, I can lay down a 5' end up here and keep on adding nucleotides to the 3' end, and it's going to keep on opening up in that direction, and so I just keep on adding on nucleotides to this 3' end until I'm done replicating this DNA. That's called the leading strand. On the bottom half of this half of the slide, again opening up off to the right, the very first time that this bubble opens, when it's just an itty-bitty bubble, I'm anti-parallel. I lay down a 5' end here, and I'm going to make DNA until I run into what was the leading strand of the other side of the bubble. Okay? And then as the bubble opens up more, I have to back up, lay down another 5' end to the 3' end. As it opens up more, back up, lay down another 5' end to the 3' end. These are the Okazaki fragments. After these are synthesized, the RNA will be replaced by DNA, and then you'll fuse these together. It takes more steps. This is called the lagging strand. Right here, at the place where the bubble first opens, the origin of replication, is the demarcation for what will be the leading or lagging strand on either side. Okay, so this defines where you can lay down your first 5' end, and because I'm opening up in this direction, this will always be the leading strand there, because it's anti-parallel to the template underneath it, and then the opposite side will be the lagging strands, and because this side of the origin of replication off to the right, it'll be the opposite orientation. The leading strand is on top, lagging strand is on the bottom. Okay, now we're going to look at that as I draw it out for you on my fancy light board. Okay, here is my fancy dancy light board that I'm going to use to explain forks and bubbles. Okay, so let's say first with a fork, okay, we're going to use the same or similar colors to the book slides, in that we have your original strand as, let's say, 5' end, 3' end, 3' end, 5' end, and I am saying that the fork is opening up in this direction, so that it's getting bigger in that direction, and I'm going to make some DNA. So first I need to lay down my primer. It's going to be an RNA primer, and we're going to put that RNA primer here, and it's going to have a 5' end here and a 3' end there. Okay, going like that. Okay, and then I'm going to make a replicated piece of DNA, and I know that my DNA is opening up in that direction, and I can only add on to DNA on the 3' end. So I'm going to make my DNA added on to the 3' end, and here's going to be my 3' end and my 5' end. And so DNA polymerase is going to add on DNA in that direction, and as the fork opens up in that same direction, I can just keep on going, and this would be called the leading strand. Okay, in that color there. Now, on the other strand, here's my 5' end right here. Okay, so that my replicated strand is going to have my 5' end here. Okay, and it's going to have its 3' end here, and first I lay down an RNA primer. Okay, and then I'm going to add my replicated strand, 5', 3', and now I'm at the end of the DNA right there. And so as this opens up, I'm going to have to do this same thing further back on the strand, and that's going to be making DNA in fragments. So this is called the lagging strand, and each one of these is going to be called an Okazaki fragment. Okay, let's now do a bubble. We're going to call this, again, this blue is our original template strand. We're going to say 5 prime here, 3 prime here, 5 prime here, 3 prime here. Right down the middle is our origin of replication. And this part of the bubble is opening up in this direction. And this part of the bubble is opening up in this direction. Okay? So again, with the primer, okay, your RNA primase is going to lay down an RNA primer, and it's going to do that in an anti-parallel direction, okay, like so, right at this origin replication, and it's opening up in this direction, and therefore I can make my replicated strand go in this direction, and this will be the 3' end, complementary to this 5' end, and as it opens in that direction, I can just keep on adding to that 3' end, so this would be considered leading. Okay, and on the other half of this strand, again, a RNA primer, 5', okay, and as the bubble first opened, when it was a little tiny bubble, the first RNA primer would have been right here, 5', 3', and it would have made a little tiny fragment of DNA, 3' right there. And this would be an Okazaki fragment, and then maybe there's another one as the RNA or as the DNA opens up more, the RNA primase lays down a primer back up here upstream, and then DNA polymerase connects this fragment with that fragment, 3', 5'. And so these would be Okazaki fragments. And this would be the lagging strand. Okay? On the other side, we have the, this is now the, if that's the 5' end, this is going to, when it was first opened, the small bubble would have laid down an RNA primer here, 5', 3', and then a Okazaki fragment, like that, 3'. As it got more open, another RNA primer here, 5', 3', and another Okazaki fragment, 5', 3'. So on this half of the bubble, this right here is the lagging strand. Okay? And this primer here that gets laid down by primase, 5', 3'. Now it is pointing in the direction that is opening here. Okay, it's opening. And so the DNA gets made in the same direction as the bubble is opening, 5', 3'. And as the bubble opens up in that direction, You just have to keep on adding to that 3' end. So for this part of the bubble, this is leading. Okay? So yes, you're going to have to be able to do this on a test where you would label every single strand that's on here, what the 5' and 3' end is, and in a bubble that is opening up in two directions, what is the leading and the lagging strands switches, both from top to bottom and on either sides of the origin of replication. Leading and lagging strands and a lot of the rules for how DNA is replicated has been figured out in bacteria. Bacteria are much easier to study than eukaryotic cells. They grow faster. They're easier to grow, but they don't have the same form of DNA. Their DNA is circular instead of linear, and they use what's called a rolling circle method, where you have an original double-stranded ring of DNA. There's an origin replication, and a replicate strand is made same way, semi-conservative, half old, half new, and the two circles roll apart from each other like so, and you get two separate chromosomes that go to two separate cells when the cells go through binary division. So we're not going to talk much more about prokaryotes other than some of the enzymes in DNA replication are better characterized from prokaryotes, but we'll get into that later. All right. So in a eukaryotic chromosome, which is linear, and it's a big, long piece of DNA, tens of thousands of base pairs long, then it's not that you start one replication bubble in the middle and then it moves down the chromosome. You start thousands of replication bubbles all at the same time, and they will move and fuse into each other until you end up with a replicated chromosome. So this is original chromosome here to dark blue, and you end up with a replicated two chromosomes that is half old and half new. Okay. If you didn't do this, open up multiple replication bubbles and do it all at the same time, with the speed at which DNA polymerase copies DNA in eukaryotes, it would take over a month to copy just one chromosome. And you probably have some sense that our cells divide faster than that. And of course, you need double your DNA before they divide. And so it can happen much faster by opening up multiple replication bubbles, start DNA replication at all those bubbles, and then as the leading and lagging strands meet from those bubbles, they will fuse and you'll end up with a completely connected piece of DNA on the leading strand and the lagging strand, and two replicated chromosomes as a result of that, and much faster than it would be if you just started in one place and went all the way to the end. Okay, what is the place to start? It's called the origin of replication. In eukaryotes, there are proteins associated with the origin of replication. These proteins are called ORC and MCM. ORC stands for Origin Recognition Complex. MCM stands for Mini Chromosome Maintenance Protein, and then a helicase loader. Helicases unwind the DNA. These will recognize the origin of replication by its sequence. There are consensus sequences of DNA that indicate the origin of replication. If you are, let's say, AAA, TT, GG, GC, GC, that's going to have a particular shape in the DNA strand. That shape is recognized by the origin recognition complex, which then recruits the MCM and identifies the origin replication, opens up a replication bubble. And as we just said, this is going to happen thousands of times along a chromosome. Multiple origin and replication bubbles be open at the same time, all of them duplicating your DNA at the same time. Here's an H5P question for you. Oh, in order for you to answer that question, you have to think about this, in that bacterial cells in their rolling circle have one origin replication, whereas eukaryotic cells in their linear chromosomes have multiple origins of replication. Okay, now you should be able to answer this H5P question. Okay, here's a big long table, and this just gives me an opportunity to talk about some of the proteins that we're going to be talking about for the rest of this podcast. So there are lots of different types of DNA polymerase. Up until this point, most of the classes you've just talked about the enzyme DNA polymerase as the enzyme that's going to make DNA. Like if you're talking about PCR reaction, you'll talk about Taq polymerase as a DNA polymerase that's going to make DNA. In cells, there are different kinds of DNA polymerases. There's Pol1 and Pol3 in bacteria. The eukaryotic version of these, there's an alpha, gamma, delta, and epsilon DNA polymerase. They have slightly different functions. There is primase that is going to lay down an RNA primer, which is a short sequence of RNA that after the bubble opens up will make a part of that bubble double-stranded, and then that is what the polymerase will recognize as where should I start. So the primer tells it where to start. In this case, the primer is RNA. This happens inside your cells. When you've talked about primers before, you probably talked about them in the polymerase chain reaction or PCR, in which case they're DNA. RNA is much less stable than DNA and harder to make and keep around, whereas DNA is very stable. So the primers that are used in polymerase chain reaction when they're ordered from a company are DNA because they're cheap and they're very stable. You can get them freeze-dried and just send them in the regular mail, and they last for decades, as far as we know. DNA helicase. These are enzymes that are going to unwind the double-stranded DNA, which needs to happen in order to open up a bubble. And then we're going to talk about these clamp and binding proteins. We talked about topoisomerase type 1 and type 2. Type 1 makes a single-stranded cut. Type 2 makes a double-stranded cut, and this is to unwind and undo supercoiling in DNA. DNA gyrase, we're not going to talk much about. DNA ligase is an enzyme that is used to put the sections of DNA back together, to a DNA back together with DNA. A version of this is what's going to happen after you're replacing the RNA primers in your lagging strand and in your leading strand, and then ligating all those pieces together of DNA together to make one continuous strand. Telomerase we'll talk about, and then the RNases we're not going to talk about for a while. Okay. So here's DNA polymerase. It wraps itself around DNA, reads the template strand in dark blue here, and then adds nucleotides to form the replicated strand on the 3' end. And so if this is the 3' end, here, this end is getting longer as DNA polymerase is doing its thing. As it's doing its thing, it may make a mistake. Okay, so here's the template in dark blue. Here's the strand it's going to make. It's reading this template and says, okay, there's an A here. I need to add a T here. And then there's a T here. I need to add an A there. There's a T here. Oops, I added a G here instead. There is the ability of this enzyme to cut that mistake out and then go back and try again. This is called exonuclease activity. Okay, so as a mistake is made in the nucleotide that's added onto the 3' end of the replicated strand, then that will bend away from the binding site of the polymerase. It'll be cut by the exonuclease activity of this enzyme, which is physically in a different part of the enzyme, and then go back to the original spot to take another shot at making the correct call. So different polymerases from different organisms have different error rates. The common polymerase from Thermus aquaticus, the one is used in TAC, makes a mistake about every 1,000 base pairs. Other polymerases from other organisms are more or less mistake worthy, and this is something that you can use depending on how important it is that you have exactly the right sequence when you're amplifying DNA. Of course, your cells don't ever want to make a mistake, but then in future podcasts we're going to talk about what happens when mistakes are made, and this is one of the things that can happen, is the enzyme that is making the DNA, actually replicating the strand, can fix its own mistakes. So this is because of this exonuclease activity. Okay, here is a H5P question. Okay, I'm going to walk you through this figure. There's just more detail at the replication bubble. Okay, so at an origin of replication, an initiator protein will bind to the double-stranded DNA at the origin of replication, and using ATP will start to unwind that DNA. So just opening up the bubble, okay, at the origin of replication. Then new proteins are going to come in. DNA helicase will bind to the unwound DNA and continues to do the unwinding. So the initiator protein got it going, and then helicase will keep it going. DNA gyrase performs strand separation by inducing negative supercoiling in front of the DNA helicase. So this is unwinding, and this is doing the opposite of unwinding. I said we weren't going to talk about this, but we are talking about it right now. Okay, and then keeping these strands apart are these single-stranded DNA binding proteins, SSB, because these are going to want a hydrogen base pair back together. So you got to keep them apart in order for them to act as templates for the new strand of DNA that you're going to make. Oops. Then primase is going to lay down an RNA primer. This is the only place you're going to see an RNA primer is in DNA replication. In transcription, where something similar is going to happen, you're going to make RNA from DNA. There is no primer. So DNA replication needs a primer in order for the polymerase to know where to start, and primase lays down that primer, and the primer is RNA, shown here in red, using the original strand as a template. So these would be RNA nucleotides that are complementary to the DNA here. So if there's an A on the original strand, there would be a U on the RNA. If there's a C on the original strand, there would be a G on the RNA, and so on. Then DNA polymerase 3, if this is bacteria, it's polymerase alpha. If it's a eukaryote, followed by delta or epsilon, that's going to start actually making DNA. Okay, and this is opening up in this direction, so this would be the leading strand, and it's complementary to the template strand, so 5' on the left hand side. This is the 3' end. You're going to keep on adding nucleotides to that 3' end as we go forward. Okay, that's where we were right there. Okay, this is just the rest of the slide, so we just did 4. Now on 5, now we're going to look at the lagging strand. In the lagging strand, again, our RNA primase is going to lay down an RNA primer once it opens up enough to do that. And it's going to add on nucleotides to the 3' end of that RNA primer, and it'll add nucleotides in the direction that is opposite to the direction that the strand is open, that the bubble is opening up. Okay, here it's drawn as a fork, but they're just not showing you the other half. Okay, and then this will continue as the bubble opens up more. The leading strand has no problem. You just keep on adding on to the nucleotides on the 3' end of the leading strand, and the RNA polymerase is going to keep going in this direction, and that's the direction that the bubble is opening. But for the lagging strand, it's going to have to add this is the original Okazaki fragment, and now it's opened up more. So the RNA primase is going to back up here and lay down a new RNA primer, and then add to the 3' end of that RNA primer, and DNA polymerase will work until it runs into the first fragment, okay, the one that was started up here. Okay, now this is opened up more, and the second fragment is going to run into the first fragment, at which point DNA polymerase will fall off. A new primer will be laid down, and DNA polymerase will rebind back here on this new primer, and then make DNA until it runs into Okazaki fragment number 2. And then number 4 happens, and then so on. Eventually, what's going to happen is that the RNA primer is going to get removed on the leading strand and on the lagging strand. It gets removed by the exonuclease activity of DNA polymerase, okay, and it's replaced with DNA polymerase activity of also DNA polymerase. So DNA polymerase will go back, another copy of it, replace that RNA with DNA. And then any fragments that are left over, such as in the Okazaki fragments that have to be connected after the RNA primer gets replaced by DNA polymerase, they're going to be stuck together by DNA ligase. Okay? And eventually what you'll have are 2 completely whole strands that have been replicated that are anti-parallel to the original template strands, and then each strand will be half old and half new. Okay, this is a really complicated looking diagram, and I'm going to break it down for you, and it's not that bad. So in this sort of diagram, it has DNA polymerases all over the place. The reality is they're actually all bunched up in this giant complex called a replosome. So you can hopefully recognize DNA polymerase. Here's one here, and here's one here. They're synthesizing DNA in the opposite direction, but they're actually very close to each other. They're held together by this clamp loader and this tau protein. So this whole structure is held together by this very cool looking structure that includes a clamp and tau protein. What the clamp is a donut shaped protein that is holding the DNA next to the polymerase, so that the polymerase can keep going. So it's like it's feeding it this thread through the eye of this needle, keeping the DNA on the polymerase so it can keep going. So here's the leading strand. Here's the 3' end that it is making. And the DNA ahead of the leading strand is being unwound for it by DNA helicase. And so it can keep going as it moves in this direction. So the template will be unwound for it, and it can synthesize new DNA, and because it's going to stay stuck on that DNA because it has a clamp next to it, okay, and it's going in this direction. On the lagging strand, the DNA is bent around in a circle, not really a circle, but a loop. This is known as the trombone, like the slide of a trombone. It's slid out in front of it, okay? And it is making DNA in the opposite direction. So if you notice how these things are oriented, how this sort of catcher's mitt shape is oriented, the thumb of the catcher's mitt is pointing that away because that's the way that it's synthesizing DNA, and it's pointed that away because this is the way that this one is synthesizing DNA. So in this picture, what is happening is there's a clamp, just like there's a clamp on here, keeping the polymerase on the DNA, and it's synthesizing that on the 3' end of this fragment. Okay, it started down here on an RNA primer, and it's made this part of the Okazaki fragment, and it's not quite done. Here's the 3' end, and it's going to keep making DNA until it runs into this Okazaki fragment, which has already been made, okay? And then it will stop. And when it runs into this one, which was made previously, it'll fall off. And the clamp will also fall off. And then it'll need to be reloaded on this new Okazaki fragment that is just starting to be formed, okay? And the clamp loader has a spare clamp ready to go when polymerase wants to load onto this new Okazaki fragment that is ready to go, and it will load on a new clamp. So here was the original Okazaki fragment. Here's the one that's being made now, number 2. Here's the next one, number 3, that's going to be made. And to get from number 2 to number 3, the polymerase has to fall off, which means the clamp is going to fall off. But then the polymerase is going to grab onto this new one, and it's going to have a clamp loaded onto that by the clamp loader, which has a spare one ready to go. And that's how the lagging strand is going to work. So this is just a more realistic view of what the arrangement looks like at a replication bubble. It's not that you have a polymerase over here and a polymerase over there at different parts of the strands. There are definitely multiple copies of the polymerase, but where the DNA is being unwound and synthesized, it's being synthesized in both directions right next to each other just by looping the lagging strand around in this trombone sort of structure. All right, because of how this works, how we've described about how the DNA replication works, where you lay down an RNA primer, and then later on the DNA polymerase will excise that RNA primer and replace it with DNA. It ends up with a problem in that at the very end of a linear chromosome, you're going to end up with a 5' end that has its RNA primer taken out, and then there's no 3' end left over to fill in the gap. So you end up with this RNA primer being released, and then there's no 3' end over here so that I can add on some more nucleotides and fill this in. So normally what I would do is I'd lay down a new primer and then I'd use the 3' end of that primer to fill that in, but there's just not enough space to do that at the end of a linear piece of DNA like in eukaryotes. And so the real consequence of this is that you end up with the ends of chromosomes, which is made up of telomeric repeat DNA. They get shorter every time the DNA is replicated. And this is thought to be one of the reasons that we age, is that for most cells, they can go through about 50 cell divisions until their telomeres get so short that it eats into the non-telomeric parts of the DNA, and then the cell dies. Can't be replicated again. Well, there is an enzyme called telomerase that can add on to these ends, but the telomerase is not active in all tissues. So how telomerase works is it contains within it both protein and RNA, and the RNA within the protein is a template for these telomeres. So telomeres are repeating regions. In this case, the repeat is TTGGGG, and that was the end of the DNA. And here's the 5' end that is sort of hanging off there, and the 3' end hangs past it because here's where that RNA primer would have been excised, leaving this hanging out there. So telomerase uses its own RNA to base pair with some of the hanging off DNA, T with A, T with A, G with C, and the rule that is a universal rule is that you can always make a nucleic acid longer by adding on to the 3' end. And so telomerase adds onto the 3' end using itself as a template, laying down another repeat, and it may do it again, and it may do it again, so that this 3' end gets longer and longer. And now there's enough physical space for the regular old way of replicating DNA to work. That is, you lay down a primer, an RNA primer, and then that provides a 3' end for which DNA polymerase to come in and fill the gap. So once this gets long enough, you lay down an RNA primer, and then DNA polymerase will fill in the gap, and it can replace some of the length that is lost from a telomere at the end of replication. So telomerase in multicellular animals is only found in gonads. Those tissues go through multiple rounds of meiosis, and in that cell division you could get very short telomeres, and so in gonads you have telomerase activity to get around that problem, but you don't have it in somatic cells, body cells. Normally, telomerase can be activated in cancer cells. So cancer cells that go through multiple uncontrolled cell divisions would have the same sort of problem in that their telomeres would really get short. So they solve that problem by activating telomerase. So they are effectively immortal, that they don't have this Hayflick limit of 50 cell divisions and then they're done. And this question about if the telomere limitation is why we get old was the motivation behind the cloning experiment of Dolly the sheep. So Dolly the sheep was the first mammal that was cloned successfully. And the technique was called somatic nuclear transfer. In the case of Dolly, they took an adult 6-year-old sheep, and they took a nucleus out of its mammary cell and took that old 6-year-old nucleus with short telomeres, and they implanted it into an egg that had its own nucleus taken out. And then that egg was artificially stimulated to start dividing in being carried by a surrogate mother carrier until Dolly went to gestation, and then Dolly was born. And the idea was to see if Dolly aged prematurely. Um, she lived to, I think, 6 years old. I can't remember that exactly, but she seemed like a normal sheep. After the fact, looking at her DNA, there were more breaks in that DNA, unrepaired breaks, than is typical for an animal of that age. Um, so there's still controversy about whether or not the telomere theory of aging is valid. Most people in the aging field that I know, that's not their favorite hypothesis about why we age, but it's still an interesting idea.